Hello guys, let's look at fertilization in flowering plants. And fertilization is a process by which the male gametes fuse with the female gametes. And in flowering plants, as soon as pollination occurs, pollen grains absorb some nutrients from the stigma and develop an outgrowth known as the pollen tube. Remember, we said pollination, the pollen grains will land on the stigma, and the stigma produces a sticky substance which enables the pollen grains to stick, and they absorb the nutrients from the stigma cells or tissues and develop into an outgrowth called pollen tube. And before we explain the process of fertilization, we must know the structure of a mature pollen grain. And the mature pollen grain, it has a tube nucleus, which is here. It also has a generative nucleus, which is here. And these two, the tube nucleus and the generative nucleus, will be of importance in describing the process of double fertilization in flowering plants. Then the inner layer, we call it inner layer of the pollen grain, we call it intine, where the outer layer, we call it exine. And this pollen grain, when it absorbs nutrients, it develops into a growth or an outgrowth we call pollen tube. And before we look at the process, we must also know in the ovary we have what we call an embryo sac, which has the following components, that is antipodal cells, the polar nuclei at the middle, then egg cell, then we have the synergids. We always use a mnemonic called EPS, and the EPS is for antipodal cells. P stands for the polar nuclei, 2. I said here the antipodal cells. Then E is for egg cell here. Then S is for synergids, that is a mnemonic used to remember the contents of the embryo sac. Remember, we must know those contents because they are important in explaining the double fertilization. You can look at this diagram. The pollen grains have landed, formation of a pollen tube, which eventually enters the embryo sac through the micropyle. We have the embryo sac here, and the contents of the embryo sac are here. We have our ovary. Let's look at double fertilization in flowering plants. As we have said earlier on, when the pollen grains land on the stigma, they are able to stick due to a sticky substance secreted by the stigma, and they obtain nutrients, and the pollen grain develops into a pollen tube. You can see our pollen grain here with a tube nucleus. Then we have the generative nucleus, and this pollen tube is developing into a pollen tube, as you can see here. Let's continue. The pollen tube grows down the style to the embryo sac, taking along with it the male gametes. And the growth of pollen tube into the stigma through the style to the ovary is by pushing itself between the cells, where it gets nourishment from the style tissues. And during this process, the tube nucleus are carried at the tip of the pollen tube. This is the tip where we have the tube nucleus. Then the generative nucleus is just behind the tube nucleus. Somewhere here we have a generative nucleus, and the generative nucleus divides by mitosis to form the two male nuclei here. As you can see, the two male nuclei, this one and this one, and we are saying the generative nucleus here which divides to form these two male nuclei as the pollen tube grows down the style. The pollen tube enters the ovules through the micropyle and penetrates through the embryo sac where it bursts open. And this tube nucleus disintegrates and one male nucleus fuses with the egg nucleus to form a diploid zygote that later becomes the embryo. Then the second nucleus, the second male nucleus, fuses with the polar nuclei to form a triploid primary nuclei, which later becomes the endosperm, denoted by 3N because there are 3. And this is the process of double fertilization. And this process is unique because it involves two male nucleus fusing with the polar nuclei and the egg cell. I'll take you back for you to understand more. In short, this generative nucleus divides by mitosis to form two male nuclei, and the two male nuclei fuses here with the polar nuclei to form a triploid primary endosperm, which later becomes the endosperm. Then the other male nucleus fuses with the egg cell here, fuses with the egg cell to form a diploid zygote that later becomes or divides to form the embryo. And this pollen tube, the pollen tube you are seeing, the pollen tube enters the embryo sac via the micropyle here, and upon entering the embryo sac, the tube nucleus disintegrates, paving way for the two male nuclei to enter the embryo sac. And that is the process of double fertilization. Let's look at what are the changes that occur in a flower after fertilization. And number one, fertilized ovules will develop into seeds. Then external protective parts and the petals wither and drop, e.g. calyx sometimes may persist or dry up or fall off. Then also the male parts like the stamen, filament style will dry up. Also the corolla will dry and fall off. Then the ovary and the ovules start developing, and the seed coat becomes, the integuments become the seed coat of the tester. The triploid zygote becomes the endosperm. The ovary wall becomes the pericarp, and the pericarp is made up of mesocarp, endocarp, and the epicarp. Then maybe the ovary becomes the fruit. Those are the changes after fertilization in plants, that is the flowering plants, and the double fertilization process. I hope you enjoyed. In case of any question, you are free to ask in the comment section. You can like and share widely. Thank you.